Hi, in this video, I'm going to go over chapter 3, which covers cell structure and function, and this is for CSUN's Bio 315 lecture. All right, so this is the outline for the lecture. We're going to basically go through exactly how the book has formatted it. So we're going to talk about the bacterial cell as an overview, then we'll go over the different parts of the bacterial cell, starting with the cell membrane and cell transport, then the envelope and cytoskeleton, cell division, cell polarity, different kinds of structures, vesicles, nanotubules, and then other types of specialized structures as well. And keep in mind that we are going to be talking about cell division here, but we're just going to go over it very briefly. There is a whole nother chapter where we talk about DNA replication and cell division as well. So this kind of just gives you an overview of bacterial cells. Okay, so let's start with 3.1, which is the bacterial cell. We're just going to go over a quick kind of overview of everything that's going to happen. So bacteria are prokaryotes, which means that they don't have a nucleus. And a lot of people think that bacteria are very simple organisms, but actually they're quite complicated. And yeah, they've got a lot going on. And so here is just kind of an overview of what you can see inside of a bacterial cell. As you can see, it's pretty packed. It's always funny when I look at biology books and they show a eukaryotic cell, there's like so much space. But in a bacteria, it's very packed. There's a lot going on. So yeah. So things that you're going to find obviously is ribosomes. So bacteria need to make proteins. So they've got ribosomes. They've got polymerases like RNA polymerase, DNA polymerase. So they've got a bunch of proteins. They've got some chemoreceptor arrays or molecules that kind of detect certain molecules outside of the cell. So we'll talk about that. They have ATP synthases to make ATP. So, they've got a lot of the same components that you would see in a eukaryotic cell, but obviously they're quite different in terms of their structure. And so one of the main things that's different is the cell membrane and their cell wall. So their kind of outer layers are slightly different than a eukaryotic cell. By the way, this is just what is in here. Obviously, you don't need to know all of this, but this is just some examples of what you might find: a bunch of proteins. Again, ATP synthase transporters, so pretty typical for a cell. Okay. And then there's a table. Again, you don't need to know this. You don't need to know the numbers or anything like that. But just to give you an example of what's in there, obviously water, like all cells, got a lot of water in their proteins, makes up a lot of them. They've got RNA. Oh, by the way, this is the percentage of their weight. Again, you don't need to know this, but like all cells, they're about 70% water. So that makes sense. Proteins. Then they have some RNA, DNA, lipids, bunch of stuff. So that's pretty typical of a cell, but this is specific for bacterial cells. Okay, and don't worry about, specific types of bacteria. We're going to talk about specific types of bacteria later in the class, but this is the classic E. coli, which is like the model organism for microbiology. And they're looking at this cell in exponential growth. So you haven't learned what that is yet, so don't worry about it. Okay, so that's pretty much it for 3.1. I just wanted to kind of briefly go over that. So now we're into 3.2 in the chapter, and we're talking about the cell membrane. So the cell membrane is synonymous to the plasma membrane. You might have heard it that way in your cell bio or gen bio classes. So plasma membrane, cell membrane. Now in microbiology, we primarily call it the cell membrane. I don't really know why, but that's just why. That's just the thing that we stick to instead of calling it the plasma membrane. But just like the plasma membrane, the cell membrane is selectively permeable, meaning that certain things can go through, but not everything. And again, it's that barrier that surrounds the cell. So you're probably familiar with the cell or plasma membrane from previous classes. Of course, the whole point of the cell membrane is to separate the outside from the inside. So the cytoplasm, which is on the inside, is separated from everything else through this cell membrane. Okay, and as you can see, just like the cell membrane in eukaryotes, very complicated. There's a bunch of proteins and other things involved. So it's not just the phospholipid bilayer. Okay, so let's talk about these membrane proteins. Like I just said, it's made up of, sorry, not membrane proteins, membrane lipids. So like I said, it's phospholipids, just like in a eukaryote. So phospholipid bilayer, 2 fatty acids, and a phosphate head. That's what a phospholipid is. So here's your phosphate head, and then you have your 2 fatty acids attached to it. And in the cell membrane, it's a phospholipid bilayer, meaning that you have 2 layers of it. By the way, in the textbook, they call each layer a leaflet. So one layer of phospholipids is one leaflet, and then the second layer, the inner layer, is your second leaflet. So that's just how they call it. So just some examples of phospholipids that you'd find in bacteria. So phosphatidylethylamine. It's a phospholipid with an ethanol amine group, so that's here. And as you can see, the ethanol amine group is positively charged. Okay. Then there's another one, phosphatidylglycerol. That one has a glycerol group. I don't have a picture of it, but the glycerol is negatively charged. Anyway, so those are just examples. You don't need to know those specifics, but those are just examples of some phospholipids that you might find in a bacterial cell. Okie dokie. And then of course the cell membrane also has these proteins called membrane proteins that are embedded inside. So some reasons why they might have it: structural support. So because there is not really organelles inside of the bacteria, as you know, since they're prokaryotes, they don't really have membrane-bound organelles, as you would call them. So there's no ER, there's no Golgi, there's not really any compartments. And so everything's kind of just swimming around. And so there needs to be some structural support, or else everything is just floating around like crazy. And so some things actually anchor to structural proteins on the cell membrane to keep them in place. So that's one reason why a bacteria might have a membrane protein. Another reason is to detect the environment. Obviously, bacteria, live in different types of environments, different pHs, different temperatures, all of this stuff. They live everywhere on Earth. And so bacteria, it's very important for them to detect changes in the environment. And so they can use membrane proteins to do that. And we'll obviously talk about specifics as we go along in the class. These are just examples of some reasons why they might have a membrane protein. Secretion and communication. So some bacteria, as you know, are pathogenic, and so they'll export toxins, or maybe they want to bind to certain signaling molecules to communicate with each other. Lots of different reasons. And then also for transport, obviously, to move ions and other things in and out, and also to store ion gradients. So a lot of bacteria, they use ion gradients as one of their ways to use an energy form. And so they can control whether or not ions are moving back and forth to create a concentration gradient, which then they can use for energy later on. So pretty cool. Okay, like I said, we're just kind of doing a brief overview. We'll obviously go over very specific types of membrane proteins as we go through the class. Okay, membrane transport. I didn't want to go through this in too much detail because you should be able to remember a lot of membrane transport from your general cell bio classes or just general biology classes. But this is something that is, I guess, unique in bacteria, is that certain things can pass through the membrane and certain things can't. And so things that can pass through the bacterial cell membrane are uncharged acids and uncharged bases. So basically, acids and bases that don't have a charge are able to pass through the cell membrane of bacteria, and things that cannot pass are going to be charged acids and bases. And remember, maybe you learned this in chemistry, that acids and bases are in equilibrium. So you have the uncharged version and you have the charged version. So it's only the uncharged that can pass through the cell membrane. The charged versions that have a positive or negative charge cannot pass through the cell membrane. So that's kind of important, especially when we think about drugs. So for example, penicillin, it has a uncharged and a charged form. And so you got to think about, which form do you want? Obviously, you want the uncharged form. Same for tetracycline and other drugs. So yeah. Other things that obviously can't pass through the cell membrane are going to be very large molecules. So we'll talk about that. And in the book basically goes over passive and active transport and osmosis. Again, you should have already learned this in your Gen Bio class. So I'm not going to go over it, but you definitely should still review it if you don't remember what active and passive transport is and what osmosis is. Because, even though I'm not going to specifically test you, it might come up in, a different topic. And so you still need to understand these concepts. All righty. So when it comes to the cell membrane, because bacteria are always kind of put into different environments, stressful environments, very hot environments, things like that, they can alter their membrane lipids based on the kind of situation that they're facing. So for example, this molecule here called cardiolipin. This is the long version of it, but you can just call it cardiolipin because I can't even pronounce that. Cardiolipin is this double phospholipid. So instead of it being one head and two fatty acids, it's kind of like two heads stuck together, and then there's like four fatty acids total. So it's like a double phospholipid essentially. And bacteria typically add these to their cell membrane during starvation, and they do it at the poles. So here they're showing that cardiolipin is highlighted in red, and you can see that the cardiolipin is concentrated at the ends, at the poles of the bacteria. And they say that this cardiolipin, I guess, prevents the bacterial cell from getting too big, which makes sense because when they're starving, they don't want to grow too much. They just want to stay in the small size. So that's all the book said about it. So that's all I'm going to say. All right. The side chains for the lipids that they use in their phospholipids can also change. So for example, this is a very normal phospholipid fatty acid. This fatty acid is just a regular carbon and hydrogen chain, right? So super simple. But you can have them in different orientations. And the ones that have kinks are going to be really important because they help make the membrane more fluid. So you probably learned this in some class where the unsaturated fats are liquid at room temperature because they have these kinks, and these kinks prevent them from packing very tightly. So it's the same principle here. If bacteria need to make their membrane more fluid, they will add these side groups, which make their membranes more fluid because then the fatty acids have kinks in them. And so what would be a situation where a bacteria would want to make their membrane more fluid would probably be very cold situation. So when they're in a very cold environment, their membrane becomes more rigid, and that's not good because they want to be able to be flexible. And so they can add these side groups to their phospholipids so that the fatty acids are more fluid. Alrighty. And then the opposite of that are these hopanoids. So you might have learned this where cholesterol causes the membrane to be more rigid or stiff. And so these hopanoids do the exact same thing. They almost look like cholesterol, literally, with the rings. But anyway, bacteria can embed these hopanoids into their cell membrane, and it causes the membrane to be more stiff. So this would be a situation where maybe a bacteria is in a very hot environment, and so their membrane is already very loose. And so they add these hopanoids to keep the membrane stiff instead of them just falling apart. Alrighty, so that was 3.2 in the chapter. So now we're on to 3.3, where we talk about the cell wall and the envelope of the bacteria. So the cell wall is this rigid structure that goes around almost all bacterial cells, and it's made up of this molecule called peptidoglycan. So peptidoglycan, also called the sacculus, but I call it peptidoglycan. Well, let's break down this word. Peptido means peptide, so there's going to be some kind of amino acids or proteins involved. And then glycan stands for, well, a sugar, right? Glycan or glycogen, glucose. These are all words that have to do with some kind of carbohydrates. And so we know that this molecule is a mixture of some kind of protein or amino acid and some sugar molecules. That's pretty much what it is. And so this is the structure of peptidoglycan. It's actually one giant molecule because everything is bonded together, covalently bonded. So everything is just one gigantic molecule. Helps the bacteria maintain its shape because, again, like I said, it's this very rigid layer that surrounds the bacteria. And so the way I like to think about it is like chainmail. If you don't know what chainmail is, you can Google it, but chainmail is like a thing that they put around like knights. Okay, I'm not explaining this very well, but you know, like in medieval times, people who were like knights and the guys who fought. When I say knights, you're probably thinking nighttime. I'm talking about knights, like the ones with swords and shit. So those guys, they have chainmail around them. And so that again is like a protective layer. So peptidoglycan is very similar honestly, very similar in structure as well. It kind of looks like chain. Right? You got things that are like kind of rings attached to rings, and everything is one giant structure. So that's what peptidoglycan is. Okay. And so let's look a little bit further into the structure of peptidoglycan. So you have these alternating NAM and NAG glycans. So that's the sugar part. Remember, I told you peptidoglycan means some kind of amino acids and mixed with some kinds of sugar. So the two sugars are NAM and NAG. That stands for these two. So NAM stands for N-acetylglucosamine. Just kidding. And that's NAG. N-acetylglucosamine is NAG. And then N-acetylmuramic acid is the NAM. Okay, so that's the NAG and that's the NAM. Okay, and those two are linked to each other, just like a disaccharide would. Okay, but then there's like a bunch of them linked, alternating basically. So it's NAM, NAG, NAM, NAG, like there's alternating chains of these. Okay, so that's your first chain, and you have a bunch of these. And then those chains have to be linked to each other. And so that's going to be linked through the peptides. So there's a peptide bridge. So these bridges here, okay, are all peptide bridges. And it's basically 4 amino acids sticking out from the M, the NAMs, okay? So the peptide bridge, by the way, comes from the NAM, not the NAG. Okay, so each NAM has this peptide bridge for 4 amino acids coming off of the NAM, and then 4 amino acids coming off of this other NAM, and then they kind of meet in the middle, and then they link there. So technically, it's actually 8 amino acids, but it's 4 coming from this NAM and 4 coming from the other NAM. This is a NAM here, the purple, and this is a NAM. So there's 4 amino acids coming here, 4 amino acids coming off of here, and they kind of overlap a little bit at the edge. And so that's how the peptide bridge is formed. Okie dokie. So, yep, that's the structure of peptidoglycan. All right, so let's move on now to the entire cell envelope. So a lot of students have a hard time differentiating the cell wall and the cell envelope. So the cell envelope basically is your cell membrane, which we just talked about, the cell wall, which we just talked about, and then any other layers that are also involved. So basically, it's all of the layers that surround the whole bacteria. So the envelope is all of it. And so there are 3 types of envelopes in the bacterial world, meaning that there are, I guess, 3 groups of bacteria that have 3 different types of cell envelopes. So we're going to talk about each of the 3, but these are the 3. So we have gram-positives, which are also called the firmicutes. They have a very specific type of cell envelope structure. And then gram-negatives, which are the proteobacteria. They also have a specific type of envelope structure, and then finally the mycobacteria. So we'll talk about each of these 3 in the next 3 slides. But here's just an example of the difference between the gram-positives and the gram-negatives. So as you can see, one of the most significant things is that the peptidoglycan layer, which is the cell wall, is much thicker in the gram-positives than it is in the gram-negatives. So that's just like one difference. And again, we're going to talk about the differences in a second, but that's just something that I wanted to highlight. Okay, so let's first talk about the gram-positive envelope. So it has a cell wall. Well, first of all of them have a cell membrane. So I'm not going to discuss the cell membrane part because all of them have a cell membrane. Both the gram-positives, the gram-negatives, and the mycobacteria, they all have cell membranes. So we're not going to talk about that because that's all in common. I'm really highlighting the differences. So the cell wall in gram-positive bacteria has a very thick peptidoglycan layer with about 3 to 20 layers. So it's way more peptidoglycan than the others. And they also have these teichoic acids. So this is a teichoic acid molecule. It's a pretty long molecule. It's got a glycerol in the middle, kind of chains of glycerol actually. And basically this teichoic acid gets intertwined within the peptidoglycan layer. So let me kind of go back to this image here. You can't really see it, but here is the peptidoglycan layer. And all of these kind of blue is the teichoic acid, okay? So don't get that confused with the cross links, the peptide bridges. The peptide bridges are not, I guess they're not shown. I can't really tell in this picture, but these blue lines are not the cross bridges. Those are the teichoic acids, okay? So don't get that confused. Okay. So that's that. So it's a negatively charged molecule that kind of intertwines within the cell wall or the peptidoglycan layer. And then some gram-positives also have what is called an S layer. This is a layer even outside of this, so like it's even further out. And this S layer is usually made up of proteins or glycoproteins, but they usually help with structure and virulence. So virulence has to do with the bacteria infecting a person or whatever it wants to infect. And so this S layer usually helps protect the bacteria and helps the bacteria avoid the immune system and all that stuff. So this is an example of the S layer. It's a very like hexagon shaped, like intertwined thing, kind of similar to the peptidoglycan layer, but even more outside. And then some gram-positives even have a capsule. So a capsule is a slippery layer, usually made up of polysaccharides. Usually is also used for virulence because, makes them slippery. So they're not as easy to catch with immune cells, things like that. So, and again, with these two, we're going to talk about them in detail when we go over, how bacteria actually cause infections and stuff. So right now, just know that they exist. That's pretty much all you need. By the way, the S layer capsule are not found in all gram-positives. Only some have these two. It's kind of like an exclusive thing. Alrighty. So now let's move on to the gram-negative envelope. So for gram-negatives, they have a very different type of envelope. By the way, these are two pictures. So this is one picture. I should have put a box around them. And then this is another picture. Okay, I don't want you to think that this is attached to this. So this is all one picture up here. I'm just going to draw a line. Okay, this is two separate pictures. Sorry about that. Okay, anyway, so some key highlights of the gram-negative envelope is that they have a cell wall, just like the gram-positives do, but their peptidoglycan layer is very thin. There's usually only one or two layers. As you can see here, there's only one layer of the NAM and NAGs. So that's that. They have this specific type of protein or specific type group of proteins called the lipoproteins. And here's just an example of the murin lipoprotein. There's different types. So murin is just one type of lipoprotein, but there's different ones. And so in this case, it's murin. And it's a lipoprotein that is found kind of in the inner leaflet of the outer membrane. So before I even move on, I forgot to mention that gram-negative bacteria have two membranes. They have an inner membrane, which is your cell membrane, as we just discussed. That's the cell membrane that bacteria have. But in this case, we call it the inner membrane because gram-negatives also have a second membrane called the outer membrane. And so to distinguish the two, we call them the inner and the outer, rather than calling it the cell membrane, but it's essentially the same thing. Anyway, in the outer membrane, which by the way, that's what OM stands for, is outer membrane. In the outer membrane, the inner leaflet or the inner layer, that's where you would find these lipoproteins kind of attached to it. Okay, and again, right now we're just kind of talking about what's there. We'll talk about the function and the importance of these lipoproteins more later, especially when we talk about certain types of bacteria and how they cause infections and things like that, or how our immune system responds to these bacteria. So we'll talk about that much more later. Right now, just know that these things exist. Okay, and then on the outer side of the outer leaflet of the outer membrane, you have this specific type of molecule called the lipopolysaccharide. So lipo means lipid, polysaccharide means a bunch of sugars. Okay, and so that's to be found on the outer membrane. And then what this picture is showing, I guess I should have like labeled it a little bit better, sorry. So this is one lipopolysaccharide or LPS. LPS stands for lipopolysaccharide, by the way. So let me write that here. LPS is lipopolysaccharide. This bottom picture is zooming in on one lipopolysaccharide. That's what we're looking at in the bottom picture. Anyway, LPS is found on the outer membrane, the outer leaflet of the outer membrane, and it's the one that's kind of facing outwards. And very important about the lipopolysaccharide is that the anchoring lipid is called lipid A. So that is the lipid that is blue here that's actually anchoring it into the outer membrane. So that molecule is called lipid A. And then outside of that, the very tip of the lipopolysaccharide has this O antigen. So it's not really shown, but imagine that the O antigen would be like out here. O antigen. Again, right now, you just need to know that these things exist. We'll talk about the function of them when we talk about how certain bacteria cause disease. Alrighty, and then the only other thing I really wanted to mention was the periplasm. So the periplasm is essentially just the space between your outer and inner membrane. So when we say the periplasmic space or the periplasm, that's what we're talking about, is this space, this gap between the inner and outer membrane. Okay, cool. The last envelope that I'm going to talk about is the mycobacterial envelope. So again, its cell wall is thin, so very similar to the gram negatives, has a thin cell wall or thin peptidoglycan layer. In the, outside of that, or a little bit further away, there is these glycans. So here's the peptidoglycan, and then attached to that is just a layer of glycan. So just galactose chains, basically. And then even further out, you have these arabinins. Arabinin is a polymer of arabinose, so it's just a bunch of sugars, right? So it's just peptidoglycan. I'm sure if you licked this bacteria, it would be very sweet. I'm just kidding. But still, so peptidoglycan, very thin layer, and then outside of that is a glycan layer, and then outside of this an arabinin layer. So a bunch of sugars just, stacked up on top of each other. So that's that. But then the most important part is that outside of that, you'll find these mycolic acids. And these mycolic acids form it's not really that obvious, but they form a bilayer, very similar to the phospholipid bilayer. So all of these here, this whole chunk here, is where you would find the mycolic acids. And these are usually attached to the arabinins. So as you can see, they're like kind of all anchored to the arabinins. Let me erase that. Okay, so that is the mycolic acid. And what is mycolic acid? It's a very thick, waxy material. And these bacteria are very protected against antibiotics and the immune system because of the mycolic acid. So Mycobacterium, which is the type of bacteria that have this envelope, are typically very pathogenic. So Mycobacterium tuberculosis, for example, causes tuberculosis. So a lot of these guys cause disease, so they're very interesting. Okay, moving on to the cytoskeleton. So now we're kind of done with the envelope and all of the outer stuff. Now let's kind of move inwards and talk about the cytoskeleton. So bacteria don't really have a cytoskeleton the same way eukaryotes do, because again, they don't really have like a lot of organelles, but they do have a cytoskeleton to maintain their structure. And so there are three very important proteins that have to do with the cytoskeleton. So the first one is FTSZ. This one forms the Z ring that goes around the bacteria. So here in this circular shaped bacteria, you'll see the Z ring, FTSZ, forms the little ring that goes around the bacteria. So it kind of makes sure that they're circular. Okay. By the way, FTSZ is found in all bacteria. So all types of bacteria have FTSZ, whether they're circular or rod-shaped or whatever, they all have this FTSZ, or at least the three that I'm talking about here. Okay, the next type of protein is MREB. So MREB forms this coil. So as you can see here, it's kind of forming this coil-like structure, and that causes the bacteria to have a rod shape. Now notice that this bacteria still has FTSZ. So FTSZ is still present there, but they also have on top of the FTSZ the MREB, and that's what causes the bacteria to be rod-shaped. Again, MREB is going to be found in all of your rod-shaped cells and also your crescent-shaped cells. So now let's talk about what makes the cells crescent, though. It's Cresc or Crescetin. Anyway, so CRES, I'm just going to spell it out. CRES is this protein called crescetin, and it's what causes the bacteria to bend and causes it to have the crescent shape. So crescetin is here shown in green. It's that protein that kind of causes it to bend over. But notice that this bacteria that's crescent shaped still has FTSC and it still has MREB. So it still has the two previous proteins. It just has the crescetin on top of it, okay? So make sure that you remember that it's not just the crescetin that is present. It's crescetin plus FTSC plus MREB that makes the bacteria crescent shaped. Okay. All right, so that's pretty much it for the proteins in the bacterial cytoskeleton. And that is it for 3.3. So now we're going to move on to 3.4. By the way, if I'm talking too fast, it's because I like to keep my videos under 45-ish minutes. So if I'm talking too fast, you can obviously slow down the video. If I'm talking too slow, you can obviously speed it up. So, okay. So moving on to 3.4, we're going to talk about cell division. And again, there's a whole chapter on this, so I'm not really going to go over it in too much detail. But you should probably know that bacteria divide through binary fission, which means that when they divide, the two cells are identical. And it happens through septation. Septation just means that there's a septum that forms and the bacteria pinch in half. And so before moving on, I want you to just kind of pause the video and think, how is this similar to eukaryotic cell division? I'm sure you learned it in your gen bio and cell bio classes. But what are some things that, when you look at this image and you see the two bacteria splitting, what is it similar to eukaryotic cell division? So yeah, just pause the video and in your notes really quick, or even just in your head, just think about, what does this remind you of something that you've already learned? Okay, so that's that. So now we're gonna talk about whoops, this kind of cut off. So this should say DNA replication, my bad. So anyway, we're gonna talk about DNA replication and division. So again, there's a whole nother chapter on DNA replication, so I'm just gonna do a really dirty overview of it. So when the bacteria decide to divide, they have to replicate their DNA, and there is a region called the origin of replication. So there it is up here. And it's actually attached to the equator. So notice that the origin is stuck to the cell membrane. And this origin is where the replication begins. And basically the DNA starts to unzip here. Now, as the DNA unzips, we then, or the bacteria, they start to form what is called the replosome, which is basically just a group of proteins that are used for replicating the DNA. So DNA polymerase and all of that stuff, helicase, all those guys. They're all there. And as you can see, there are two replisomes. Okay, and the reason why there are two is because bacteria, since they have circular DNA, they can replicate in both directions. So rather than just having one direction of replication, there's two directions of replication. So that's convenient for them because then they can go much faster. Okay, and so these two replisomes basically go in different directions, and then they'll meet at the end. And when they meet at the end, they're going to reach what is called the termination site. So that red region here is the termination site. Okay, and in the termination site, it obviously terminates, so the replication stops happening there. And so once the termination site has been replicated, the cell knows that it has completed DNA replication. And so the septum forms, and the way that the septum forms is through that Z ring that we just talked about through FTSZ. Okay, so let's kind of go into a little bit more detail about how that FTSZ forms. So there is this complex called the divisome. It's just called that because it's the complex of proteins that helps with division. And this divisome is very complicated, as you can see. We don't have to go through everything, but it's this whole cluster of proteins that are involved in producing that FTSZ Z ring. And so, of course, FTSZ is involved, as you can see down here. A bunch of these FTSZ proteins form the ring that goes around the center of the cell. And then the only other protein is FTSN. FTSN is the protein that kind of regulates the constriction. So when the cell is ready to divide, it helps constrict FTSZ, so it pinches the cell in half. Yeah, that's pretty much it. We'll go into so much more detail later on in the class, so cool. So now moving on to 3.5, we're going to go over cell differentiation. A lot of people don't know this, but some bacteria actually differentiate, meaning that instead of when they divide, they are two identical cells, they divide and they are not identical. Really crazy. So asymmetrical division is something that some bacteria can do, specifically Colobacter. And that's what we're showing here, where the daughter cells are not identical. So with Colobacter, sorry, this is one image separated. I really should have boxes around them. Anyway, so here's a Colobacter. When the Colobacter divides, one of the cells becomes what is called the anchored cell. The cell is anchored and it stays there, doesn't move. And then the other one is the swimmer or the swarmer and is the one that moves and swims away. Now, why would a bacteria want to do this? Well, maybe, if you're a bacteria and you're dividing, you're probably in a very nice environment, an environment that has a lot of nutrients, whatever. And so you probably want to keep one of your cells anchored in this area that already has nutrients and you already know is a good spot. And so you keep them there. And then the other guy is the one that leaves and hopefully finds a place that also has nutrients. So one is, meant to spread and move, whereas the other one stays in this well-established environment. So makes sense that you would want to do that. Okay, so let's just briefly talk about the proteins that are involved in this whole differentiation thing. So there's one protein called TIP-N. TIP-N basically regulates where the pole, quote unquote, is going to be. And when I say pole, well, the book calls it a pole, but I like to call it the head, okay? And so TIP-N is this yellow protein here. And basically, when the cell divides, you have this TIP-N, and it always ends up in the head part. And obviously, there's no head of the bacteria, but I just like to call it the head because it seems like it's the front, even though it's not really the front. But anyway, so TIP-N kind of migrates to the ends of the heads of these bacteria, and so it establishes that this is the head part. Okay, and then there's this other guy, Pod J, and Pod J is purple, and you will only find him in the swarmer cell because it's the one that will activate the production of the flagella so that the bacteria can swim. And then finally, DivJ is the red protein, and that's going to be found only in the cell that stays put, the one that is anchored, and it's the one that produces or regulates the production of the stalk, and the stalk is what anchors the bacteria. And so that is kind of those three proteins. You don't really need to know this whole cycle of how they all move because TIP-N is delocalized and then it localizes and all this craziness. Don't worry about it. You just need to know the three proteins. Okie dokie. So when bacteria grow, there is also asymmetry, even with bacteria that are not Colobacter. So even just regular old E. coli, people have done studies and found that there is asymmetry even in just regular ass bacteria. So we call it the old pole, which is basically the portion of the bacteria that has old cell wall. And then the importance of this is that the old cell wall is more likely to degrade or rupture because it's older. Whereas you have the new pole, which is basically cell wall that has been freshly made just before cell division happened. And so there's always going to be a new pole and an old pole. And when you divide the bacteria, notice that the bacteria will get some old and some new, and then some of it is some old and some new, okay? And then every time the bacteria divide, the progeny or the daughter cells will have some old, some. And so again, I want you to pause the video and just think about, what does this remind you of? Where else have we seen this situation where after something is split, or after we have duplicated something, hint, hint, there is some old and some new, some old and some new. Where else have you seen this in biology where we've got one thing, we duplicate it, and the two daughters or the two things that you make have an old and a new and an old and a new component? Okay, like I said, you pause the video, so. All right. All right, moving on. So now we're just gonna rapid fire go over a bunch of other things that you find in bacterial cells. So we're starting with vesicles and nanotubules. So vesicles are just kind of like the vesicles that you see in eukaryotic cells, except the vesicles are more for export rather than for movement inside. So all of these vesicles are usually used to export things outside of the cell, whatever the hell the bacteria needs to move. And then this really cool thing, nanotubules, bacteria can actually create a tube that connects one bacteria to another. So that's literally what's happening here. One bacteria is literally connected to another through a tube, and they can share things with each other. Really crazy things like proteins or RNA. They can literally share different types of resources with each other. So that's something that you don't think about when you think of bacteria, because you always think of them as solo organisms that don't really do anything with each other. But here, you can see two bacteria that shared their proteins and RNA with each other. So very interesting. Okay, some bacteria have thylakoids and carboxysomes. So I'm just going over random structures, right? So thylakoids, you might recognize this from thylakoid membrane of a chloroplast. That's because there are some bacteria that are photosynthetic, for example, the cyanobacteria. And just like with the thylakoids of the chloroplast, it just increases the surface area so that you can have more surface area for photosynthesis. We also have carboxysomes. These guys are the location for carbon fixation. Carbon fixation is essentially the Calvin cycle, if you think about it. And so because bacteria can do photosynthesis, they can also do, of course, the Calvin cycle. And so they will have rubisco and these carboxysomes. And yeah, that's where the carbon fixation reactions happen. Okay, moving on to some more. I want to call them organelles, but they're not, right? They're just structures. So granules, some bacteria produce these granules. That's what we're looking at here, that are filled with things like glycogen and different types of sugars, polymers for storage. That's pretty much what it is. Pili. Pili are these very thin hair-like structures that you can see on some bacteria. And these pili help the bacteria anchor to different surfaces or move around. So that's that. And then we already talked about a stalk because we talked about it with the Caulobacter, but here it is again. It's basically like a foot, right, that anchors the bacteria to a specific surface. So pretty cool. Okay. Last thing I want to talk about for this lecture are the flagella, very important structure that's found in bacteria. So the flagella is this very large whip-like structure that helps the bacteria swim around. It's better than the pili, by the way. So pili are very small hair-like structures. They can help the bacteria move, but it's not that good. The flagella is really what helps bacteria swim. And so here is just a picture of, I believe, salmonella with flagella around it, okay? And we'll talk about the arrangement of flagella. Some bacteria only have one flagella, some of them have multiple, some of them have multiple, and the flagella are all in one side or all over the place. So flagella, it can be very, there's a lot of different variants in the flagella arrangement, but let's just talk about the general structure of a flagella. So the main protein of the flagella is called flagellin. Fantastic, whoever named that, thank you. Anyway, so that's FLIC, flick, I guess. For the bacteria to flick themselves. Anyway, so it's this protein that makes up the spiral, the actual main component of the flagella. And then the rest of the structure we're not going to really talk about yet, but I do want to talk about two proteins because they're important for powering the movement of the flagella. So where does the energy come from to move flagella? It's through proton motive force. So the movement of protons from the high concentration to low concentration. So pretty much the way that ATP synthase generates ATP. And so the two proteins that are involved are MOT-B and MOT-A. I guess MOT for motility or motor. Either one, I'm not really sure. But anyway, MOT-A and MOT-B are the two proteins that are involved when the hydrogens, the protons, move from high to low. It powers the rotor which is here, and then the whole flagella just spins. Pretty interesting. Now, how does this spinning actually help the bacteria move? That is the last slide on chemotaxis. So bacteria, when they move, typically they want to move towards something, right? That's what they want to do. They want to move towards some kind of nutrient source or whatever the hell it is. So that's called an attractant. The bacteria are attracted to that region. And so there's two ways that the flagella can rotate. So there's counterclockwise rotation. So when you look at a clock, if you're rotating it the opposite way of the clock, that's counterclockwise. And this is what causes the bacteria to move towards the attractant. So when all of the flagella are spinning counterclockwise, the bacteria move forward, basically. And we call that the walk. And then when the flagella spin counterclockwise, the flagella, notice how instead of them being nice and like smooth together, they all kind of get all crazy like this. And it causes the bacteria to tumble, meaning that the bacteria start to turn in random directions. And basically, what happens with the bacteria is they will do this weird motion where they move towards the attractant, and then they tumble, and then they move towards the attractant again, and then they tumble. And eventually they'll get to the source or the attractant that they want to get to, but it takes a while. And so the way I like to think about it is when you're playing Marco Polo, if you've ever played Marco Polo with friends in a pool. So you close your eyes so you can't see, just like a bacteria, right? They don't have eyes. And you hear your friend say Marco, and the sound is the quote unquote attractant. So the sound of your friend saying Marco means, oh, my friend is towards the left, so I'm going to move towards the left. Now, when they stop talking, then you sort of lose yourself, right? You're like, oh, shoot, I forgot which way I was going, especially when you're like swimming, right? It's kind of hard. And so then that's where the clockwise or tumble happens, because now you're just like, where am I going, right? And so the bacteria are doing the same thing. And then you hear Marco again, and you're like, oh, my friend's that way now. And so you kind of just follow your friend along. You follow the sound of your friend, but in between when they say Marco, in those time periods where there's no sound, you're basically just tumbling and not really sure where you're going. So that's the way I think of chemotaxis. The bacteria go towards the attractant, but then they stop, they kind of swim around, and they're like, where am I going? And then they hear or they sense the attractant again, and then they go towards it. And that just keeps happening until they get there. Okay, cool. So thanks for watching, and I will see you in the next video.